try to understand DNA viruses, right? So I'm going to write here DNA viruses, right? So let's just talk about DNA viruses right now. We're not going to talk about any RNA, right? So the big master rule, the big master plan is that there are three trends in DNA viruses. We can understand them very easily with using these three trends. The first trend is that they are double-stranded. Okay, they are double-stranded viruses, DNA viruses. Okay, they have double-stranded, means they have these two strands. Okay. Second, they replicate in the nucleus. Okay, in nucleus the replication happens. Third trend is that their shape is icosahedral shape. Okay, so three trends, but we have exceptions, two exceptions to be remembered. Exactly, exceptions. What's the exceptions? Exception first is parvo b19. So parvo b19, it is a single stranded DNA virus. That's the first point. Second exception is that pox virus. Now pox virus replicates in the cytoplasm, cytoplasmic. Okay, that's important stuff. So replication happens in cytoplasm, and the shape is actually different. Now the nucleocapsid is actually not a classic icosahedral shape. It is more like a tubular shape. We're going to talk about it later. Okay, so the first way we can really understand them is we can split DNA viruses into two categories, right? The first category is going to be on the shape, right? So first is icosahedral shape. Second is inner tubular nucleocapsid shape. Okay, so inner tubular on the basis of capsid. So we are dividing them on the basis of capsid. Okay, so we have two categories, isosahedral and inner tubular. So the inner tubular group is basically pox viruses. That's it. Okay, so all pox viruses, right, are just inner tubular group. Okay, so if we see any sort of cytoplasmic replication, brick shaped, I'm going to write these buzzwords: brick shape, right? We see as any eosinophilic cytoplasmic inclusion, eosinophilic plus cytoplasmic. We should think about umbilicated lesions, right? Umbilicated. That's also one big buzzword. Now we should immediately think about pox viruses at this time. Now, common pox viruses, I'm going to name two of them. First is simple, smallpox, first virus. Examples. Second name is molluscum contagiosum. That's it. So we have two viruses there, right? Simple stuff. Now let me get the photos for you. Okay, leave the photos, I'll add the photos later on. So in inner tubular we have pox, right? That's important. And in isosahedral, the shape, right, we can still divide isosahedral into non-enveloped. So I'm going to write that in red, non and enveloped. Simple. Okay, they're enveloped because enveloped, right? Enveloped because they're kind of weak. They cannot stay without the envelope. And these non-enveloped are serious viruses. You don't want to mess with them. They are very resistant. Non-enveloped, right? That's the thing. So non-enveloped are the serious viruses. Okay. Now the fastest way to think about non-enveloped viruses is that non-enveloped viruses are tougher. That's the first point. They are tougher. They are more resistant to drying. They are more detergent. They are more resistant to detergents. They are more resistant to any sort of acid, and they just survive better in the environment. Often they spread via fecal, oral, or by surfaces, right? So fecal oral route. Not gonna write that down. And enveloped viruses are quite fragile, okay, and they can be destroyed by alcohol, detergents. Drying can destroy them. I'm talking about dry them, okay, drying, okay. I'm gonna dry them. So let's talk about non-enveloped viruses first. So in non-enveloped, the dangerous one, right? We can, okay, wait. So the dangerous one, wait, let me just change that. Yep. So the non-enveloped one can be categorized into single-stranded, double-stranded, right? And in double-stranded, we can still classify them under the shape of DNA. Okay, so I'm going to make these two arrows first, second, right? In non-envelope, I told you three types. Okay, so I'm going to use the color pink because I like it a lot. We have single is 1s, single stranded, another word here is linear, okay, linear DNA, okay, that's first type. The second type is going to be double-stranded but circular, okay, so I'm going to write double-stranded, but this time it's going to be circular, okay, circular DNA, okay. And the third type, it's going to be again double-stranded, but the shape is going to change now. It's going to be stranded linear, okay. It's going to be linear again, once again, linear DNA, okay. So we have this circular type, right? We have this linear one, and then again we have a linear one, okay. So single-stranded, double-stranded, okay. So single-stranded linear, double-stranded circular, and double-stranded linear. Three types under the hood of non-enveloped. These are non-enveloped, non-enveloped, right? Now let's talk about single-stranded linear DNA first, this one, okay. So here we have bunch of viruses, right? The only one DNA that, not bunch of, the only one DNA virus that is single-stranded is Parvo B19. So I told you Parvo B19, single-stranded DNA, that's the exception, right? So here we have Parvo B19. I'm going to write the name of it, right? Parvo B19, right? Now this is very high yield. Again, only single-stranded linear DNA virus. Now the buzzwords for this is that SLAP cheek rash, aplastic crisis in sickle cell, okay, arthralgias. Why aplastic crisis? Because Parvo 19 likes to infect erythroid precursor cells in bone marrow. It likes the cells that rapidly divide because it is a single-stranded DNA virus, and it really depends heavily on the host DNA replication machinery. So it needs that really high multiplication rate, okay. So a child with sickle cell, sudden drop in reticulocytes, anemia, right? So that is Parvo B19, okay, that's important. So I'm gonna write here erythroid. I'm gonna write here which diseases I told you. A child with sickle cell, so I'm gonna write child here, child plus sickle cell disease, right? I'm gonna write drop in reticulocyte counts plus anemia. Gotta look for them, okay. And very important is the cheek rash, okay, cheek. Now it needs that precursor, erythroid precursor, for rapid multiplication, okay. So cheek rash, that is Parvo B19, okay. Now let's talk about double-stranded circular, non-enveloped, simple stuff, right? This is quite actually this, how to say, distracting me myself. Gonna wait. Goes down. Yep, that's better. Okay, so we understand single stranded. Now we talk about double stranded one. Okay, so double stranded circular DNA viruses, bunch of them. I'm gonna write the names first. First name that comes to me is papilloma virus. H human papilloma papilloma virus. Second name is JC polioma virus. Okay, and the third name right now is BK. Again, polyoma virus, right? So we have three viruses: human papilloma, JK, and BK. Now, human papilloma loves squamous epithelium. That's the truth right there. I'm gonna add that to this, the story of HPV. Now, HPV loves squamous epithelium, right? Now, strains 6 to 11 warts. 16 to 18 gonna be cervical, whatever you want to call it, cervical cancer. Okay, and after 31st, 33, and 45 are also cancer associated. Okay, so just remember that 6 to 8 is warts, 16 to 18 is cervical cancer. Okay, now simple mechanism of human papilloma is that we have two proteins. Okay, now we have E6 and we have E7. Okay, simple mechanism: E6 stops P53, and E7 stop something called as retinoblastoma. retinoblastoma. So RB gene. Okay, RB protein, retinoblastoma proteins, right? All right, proteins P. Now that means that HPV disables the two big tumor suppressors genes, right? So HPV is like HPV is a tumor suppressor. It suppresses that. It stops the genes that are very important for suppressing the tumor. Okay, so these two, they work by these two, E6 and E7. It's really tested. It's tested everywhere. Very common knowledge everywhere. Let's talk about JC. Now, JC polyomavirus actually causes multifocal leukoencephalopathy. So, multifocal leukoencephalopathy. Okay, now the cool thing about is that these patients, right, are actually immunocompromised, most of them. So we have AIDS, we have multiple sclerosis, we have neurological problems, we have demyelinating diseases, right? So JC kind of jumps around the CNS, chewing the myelin. Okay, so JC is jumps around CNS myelin problems. Okay, that's a cool way to remember it. I think it's pretty cool. Let's talk about BK now. BK right there. Okay, BK polyomavirus. It's important in transplant patients. Simple stuff. Transplant patients, right? Now, association, hemorrhagic cystitis, blood in the urine. Renal transplant nephropathy. Right, important. So JC is the brain, BK is the kidneys usually. Okay, so I'm gonna write kidneys. Okay, so K stands for kidney here. Not really, but just we really trying to remember it. So BK is kidney and JC is jump around the brain. Cool. Now let's talk about double-stranded linear DNA. Okay, so here this category is just one virus. That is adenovirus. Adenovirus, simply. Okay, now this category is just adenovirus. It does a lot of things. Okay, so a bunch of things. Pharyngitis, so pharynx. We have conjunctivitis, so conjunctivitis. We have pharyngogingival virus. Sorry, they are fever, right? So they are fever as well. Pneumonia, bunch of things. I mean, really high yield pneumonia, right? Again, hemorrhagic cystitis, cystitis. Gastroenteritis, right? Gastroenteritis. Okay, now usual vignette is gonna be kid, fever, right? Sore throat, some sort of lung complaint, right? Sore throat. And very important here is that red eye, right? And more hints that we can get is usually some sort of summer camp history. Summer camp, maybe we have some military recruit respiratory illnesses, right? Now that's the thing. Cool way to think about it is that adenovirus attacks adenoids, eyes, lungs, gut, and bladder. Okay, so that's the double-stranded linear DNA viruses, right? So that part is now covered. Non-enveloped is done. Okay, now let's talk about enveloped viruses, right? Now these enveloped viruses, now we enter this fancy code side of the DNA, right? Now we have three types of enveloped viruses. We have herpes, we have hepatitis B, and we have pox, right? Now again, the pox virus is a single-stranded pox, okay? Cool thing, we should know about it. So let me just make it. So we have herpes, right? We have herpes viridae. I mean, it's a family, right? So herpes viridae. I don't want to write the virus. Hepatitis B. Hep B, right? And we have pox viruses. So let's talk about herpes first. So all herpes viruses are double-stranded. Listen to me. All herpes are double-stranded, right? All of them, wait, let me write the properties. These are double-stranded linear DNA, okay? And they are enveloped. So linear. That's the first point. Second is all of them are enveloped. Okay. Third is they have the shape. The shape is icosahedral. Icosahedral. Fourth one, they are latent. It means they are hidden after the infection. Latent post infection. Cool, they are very cool. Now we have bunch of viruses in them, right? We have herpes type 1, herpes simplex type 2, varicella zoster, Epstein-Barr, cytomegalovirus, human herpes 6, human herpes 7, HSV 6, 7, 8, right? So let me just write it simply. What kind of viruses do we have? Human simplex virus 1, virus 2, Varicella zoster viruses, right? Epstein-Barr virus, EBV, cytomegalovirus, CMV, human herpes, HHV 6, 7, and 8. So that's the family of herpes. Okay, cool. The best way to think about herpes is where they hide. Cool thing. Okay, so HSV1 hides into trigeminal ganglia. Okay, let me write that. Let me open that up for us. Open it up. Yeah, let's keep it here. Let's go here, go here, go here. So HSV1, where does it go? Likes to hide where? Cool way. Hides into trigeminal CN5 ganglion. Okay. HSV2 hides in sacral ganglion. Okay, so both are ganglionic. Okay, now let's talk about Varicella zoster, right? VSV goes and hides where? VSV likes to hide in dorsal root ganglion. Okay, dorsal root ganglion. So that's why we have these pain on rash. Okay, we have neuropathies and we have nervitis. Okay, EBV, Epstein goes and hides where? Where do you like to hide? In B cells. What else is there? CMV, monocytes. What about these family? We have 6 that hides into T cells, actually. T cells. We have 8 that actually is associated with Kaposi sarcoma. I just remembered that one. Kaposi sarcoma. Simple stuff, right? Kaposi sarcoma. Okay. So not gonna go deep into HSV1, HSV2 because we're gonna cover them after that. Cool thing we should know is that both HSV1 and HSV2 are both multinucleated, right? Giant cells. They have a positive Zancke smear. We should remember them. Okay. They both are positive Zancke smear. Want to remember it because sometimes the question can appear as such. Varicella zoster, chickenpox. Okay, so first time gonna be a chickenpox. Wait, Varicella zoster. First is chickenpox, and then reactivation happens. It goes back, and once the reactivation happens, we call it shingles. Okay, and the complications are just post-herpetic neuralgia, Ramsay-Hunt syndrome, disseminated disease in immunocompromised patients, right? EBV, Epstein-Barr, is also called B virus, right? B stands for B. Just remember that. Classic buzzwords: splenomegaly, heterophile positive, associated with Burkitt lymphoma, right? Hodgkin lymphoma, posterior cervical lymphadenopathy. Let's talk about cytomegalovirus. Now, this is a transplant and congenital virus, right? So I'm gonna write. Cool thing about it is congenital hearing loss, what should I write? First of all, any sort of esophageal, any sort of retina, eye, right? Retina in any HIV AIDS patient, basically. Okay, that's the first thing. Any chorioretinitis, right? So congenital hearing loss, owl eye inclusions, owl, that's very buzzy. Owl eye, that's also very buzzy. HSV 6 and 7, they cause the Rosella infantum, high fever first. HSV 6 and 7 we are talking about, okay? So 6 and 6, 7. They cause what? They cause first the high fever goes up, right? And then the rash appears, right? After a few breaks, post some breaks, okay? So that's the classic vignette. Baby, right? What's the vignette? The vignette is we have a baby, has fever for 3 days, right? And then suddenly a rash, appears, and it gets resolved. That's the vignette, okay? HSV 8 is associated with Kaposi, purple vascular lesion, and again, AIDS patients as well. Now, let's talk about hepatitis B, right? Okay, let's talk about hepatitis B. What we are gonna talk. First of all, now this is a little bit weird virus, okay? Now, it's weird not because it's weird. It's weird because the qualities are quite different. First of all, it's first strand. It's enveloped. So enveloped, right? Second strand is circular, right? So circular DNA. Third, very cool. I love it. Partially double-stranded DNA. Now, this is a vignette that's present almost in all the question that is suggesting about hepatitis B, okay? So, partially double-stranded. I'm going to tell you why. Fourth is they use something called as reverse transcriptase. Right? So, that's why it behaves like a retrovirus, okay? Retrovirus is just old virus, okay? So HBV, right? HBV. Now HBV enters the nucleus, right? So I'm gonna write the story of it. So it enters. Wait, let me write it here. Enters the nucleus first, right? So it enters the nucleus. It gets repaired into fully double-stranded circular DNA, okay? So it's partially double-stranded, right? And then it's repaired. So now repaired, and we get a fully double-stranded circular DNA, right? And then the host enzymes make the mRNA, okay? So host is actually making the mRNA, the messenger RNA, right? And then the reverse transcriptase converts the pre-genomic RNA back to the DNA. And then the reverse transcriptase turns the pre-genomic RNA back into DNA. So that's pretty cool, I think, right? So we're going to talk about the antigen later on. A little idea is that IgG is just prior exposure, M is that acute infection. If we have anti-hepatitis B surface antibody, it's present on the surface, okay? It means that we have the immunity because this surface of the virus is actually taken up by APC cells, antigen producing cells, right? And so we have immunity. It means we have immunity. If we have the antigen, the E antigen, it means it's still right now the transcription is going on. It's super highly infective. The virus is right now highly infective, okay? And if we have the surface antigen, it means we have active infection right now going on, okay? Just we'll keep it later on, okay? So let's just understand. this type of viruses, right? I think that's more or less we have the idea of viruses now, right? So we have, okay, so we have how many viruses now, right? So let's try to summarize this whole thing, okay? So the only single-stranded DNA virus is Parvo B19, single-stranded linear DNA, okay? Let me open up my, wait, no, another premium. Yeah, that's the thing. So only single-stranded DNA virus, Parvo B19, only DNA virus that replicates in the cytoplasm, smallpox, right? It replicates in the cytoplasm, okay? Only DNA virus that uses reverse transcriptase is hepatitis B, okay? JC, just jumping around the myelin, is JC virus. Double-stranded circular DNA. BK, K stands for kidney here. I mean, it doesn't stand, but I mean, we think we stand. So BK, poliomavirus, it's kidney. HPV, it's the P53 and RB, E6, E7, that's important. So HPV is P53 and retinoblastoma protein destruction. Herpes virus, latent forever, like squatters in the basement, right? And adenovirus is eye, throat, bladder, lungs, right? So that's whole summary of DNA viruses. That's how the whole DNA virus is actually perceived. So simply we have capsid shape, right? Right. So let's once again go capsid, icosahedral, right? Inner tubular is pox. Pox is cytoplasmic, right? Pox, and this one molluscum, very weird name, I don't like this name. Brick, eosinophilic, cytoplasmic, umbilicated lesions, right? So inner tubular is pox, capsid, isosahedral. In icosahedral, not isosahedral, icosahedral, weird name by the way, icosahedral, right? It's a weird name, right? Now what is nucleocapsid, by the way, right? The capsid, nucleocapsid is just the capsid, that's the protein coat. Nucleo means genome, right? So here the word is icosahedral, weird, right? It's a weird name. Now icosahedral is a shape, right? And the weird shape is that let me write it down. We have these triangular shapes like this going on, okay? And they kind of make like a sphere, like a football, right? Like this. I don't know how to make 3D sphere. So that's how they look, okay? Sphere. Maybe I can, I don't know. So yeah, it looks like this, but it's 3D and it's made up of triangles, right? So that's cool, I think. So icosahedral nucleocapsid means viral capsid arranged in symmetrical spherical structure. Now why would nature give us this? Because it's super stable, it uses minimum amount of protein, and it's just efficient, right? So icosahedral shape, we have herpes, the premium, we have herpes, icosahedral, we have all these shapes, right? Adenovirus, right? Icosahedral. Now let's talk about this shape called inner tubular, right? Now you're calling inner tubular shape, right? Now the thing is, it looks like a coil, like this, and DNA, the genome actually sits inside of this like a thread. Influenza and rabies, right? They're also inner tubular shape. Okay, so the capsid protein, right? That is the capsid protein, actually wrap around the genome like a spiral, forming a tube-like structure. Okay, so I'm gonna give you a little bit of imagination. That's my genome, and the shape is going to be like this, just wrapping around the genome, right? So I hope we understand inner tubular form now, right? So yeah, that's the thing. So that's it. Iso here means equal, right? So that's it. Have a nice day, DNA viruses. That's it.